So in section 5, we're going to talk about renal regulation, and this is our long-term mechanism for controlling acid-base balance, and it's by no means quick at all. It acts very, very slowly, but it does help compensate for any imbalances we have that could be a result of maybe a different diet we have, or changes in our metabolism, or even a disease. So we did have those chemical buffers that were first on scene that acted very quickly, but chemical buffers can only tie up excess acids or bases temporarily. They don't actually eliminate them from the body. And when we talked about our respiratory mechanisms, we could use our lungs to get rid of the carbon dioxide, and but we can only deal with carbon dioxide in regards to being an acid. So why our kidneys are so important is because they can actually get rid of all the other types of acids that we are generating during cellular metabolism. So things like phosphoric acid, lactic acid, or even ketone bodies. And we call these acids non-volatile or fixed acids. Also, when we're talking about the kidneys, they can regulate the blood levels of alkaline substances and also help renew chemical buffers that we use up while we're trying to regulate the hydrogen ion concentrations in the extracellular fluid. So they help regulate the acid-base balance by adjusting the amount of bicarbonate that we have in the blood. So depending on the blood's pH, the kidneys can do one of two things. They can either conserve or reabsorb the bicarbonate ion, or generate new bicarbonate ion. If we are in a state of alkalosis, then we want to get rid of the bicarbonate ion and go in and excrete it. So when we're talking about bicarbonate and hydrogen ions, generating or reabsorbing one bicarbonate is really the same thing as losing one hydrogen ion that we have. They both have the same effect on the pH. So like I mentioned, excreting one bicarbonate ion produces the same net effect as gaining one hydrogen ion. If we look back at the equation, which again I don't expect you to memorize, that's because it's going to push that equation to the right and increase the hydrogen ion concentrations. So the renal regulation of acid-base balance really depends on the kidney's ability to either secrete or to retain the hydrogen ion. So to reabsorb the bicarbonate, the kidney must actually excrete the hydrogen ion. And to excrete excess bicarbonate, the kidney must retain the hydrogen ion and not secrete it at all. So in order to maintain what we refer to as the alkaline reserve, the kidneys must be able to replenish or reabsorb the bicarbonate ion. So our tubule cells play a huge role in this, and because they are impermeable to the bicarbonate, but they are permeable to carbon dioxide. So just like we saw with our erythrocyte, we kind of have the same thing going on. So bicarbonate can gain entry back into the body in a roundabout way by being converted into carbon dioxide. Once back in the cell, the carbon dioxide can be converted back into the bicarbonate, or it can leave the cell as carbon dioxide. But the mechanism is going to be coupled to a hydrogen ion being secreted. So for every hydrogen ion that's secreted, a bicarbonate ion is going to be reabsorbed. So there's a couple steps. The first one, I'm going to show you a picture because I think this is better. So this is what's showing you what's going on. So in step 1 right here, we have carbon dioxide combining with water in the tubule cell, which is going to form carbonic acid. So here's our carbonic acid that's being formed. This is our enzyme, which is carbonic anhydrase. So once we have our carbonic acid, remember it's very unstable, so it quickly dissociates into a bicarbonate ion and a hydrogen ion. So what we're going to do is we are going to take our bicarbonate ion, send it out into the interstitial fluid, and eventually into our peritubular capillary. so we can produce more buffer, which is what we wanted to do. And we take our hydrogen ion from the split or dissociation of carbonic acid, and we actually are going to push it out into our filtrate. And our filtrate is then going, in our filtrate, the hydrogen ion is then going to form with a bicarbonate ion and reform carbonic acid, at which point we can again take carbonic anhydrase, split the bicarbonic acid into water and carbon dioxide. Carbon dioxide enters back into our cell, recombines with water to form carbonic acid, and then we're doing the same thing over and over again so that essentially we can generate new bicarbonate ion and we can recycle the hydrogen ion in order to generate new bicarbonate ion. So we do have ways of generating new bicarbonate ions. So when we break down our food, we are going to generate new hydrogen ions. And all these new hydrogen ions are going to have to be balanced by a bicarbonate ion. So we have, we secrete hydrogen ions are going to be used to reclaim the filtered bicarbonate. It's not really excreted from the body. It's going to be incorporated into the water molecule like we saw, which can be absorbed. So the body has the same number of bicarbonate ions and hydrogen ions as it started with, and we have no net gains. So it helps restore the bicarbonate ion concentration in the plasma that exists at that time. So we do have two mechanisms in our proximal convoluted tubule and our type A intercalated cells that can actually generate new bicarbonate ions by getting rid of hydrogen ions. So we have two ways in which we can do this. We have the excretion of buffered hydrogen ions, and we have ammonia excretion. Both of these are going to involve renal excretion of some sort of acid via secretion or excretion of a hydrogen ion or an ammonium ion in the urine. So let's start with the first one, which is going to be via excretion of a buffered hydrogen ion. So the binding of hydrogen ions to buffers in the filtrate is going to help lower the hydrogen concentration gradient. And this is going to allow the proton pumps of the type A intercalated discs to secrete huge numbers of hydrogen ions that the body has to get rid of in order to prevent acidosis. So the most important urine buffer is the phosphate buffer system, specifically the weak base, which was monohydrogen phosphate. So the components of the phosphate buffer system filter freely into the tubules, and about 75% of the filtered phosphate is going to be reabsorbed. But the reabsorption is going to be inhibited if we're in acidosis. So the buffer pair becomes more and more concentrated as the filtrate is moving through those renal tubules. So when we're talking about the type A intercalated cells, those are going to secrete the hydrogen ions actively via an ATPase pump. And the secreted hydrogen ions are going to form with the hydrogen phosphate. So let's look at an image. So we have our monohydrogen phosphate right here that's combining with a excreted hydrogen ion here, which is going to form our hydrogen phosphate that's going to be released out into our urine. So where do we get that hydrogen ion from? Well, we are taking the carbon dioxide that's being produced from cellular metabolism occurring in the cell. Remember, anytime we're generating ATP, one of the other products is water. And we get carbon dioxide and we get ATP. So the basically cellular metabolism that's occurring in the mitochondria in these cells produce carbon dioxide and water, we have the enzyme carbonic anhydrase to help us form the bicarbonic acid. We still have the same thing where carbonic acid is not stable and is going to readily dissociate into a hydrogen ion and carbonic acid. And then our bicarbonate ion is going to go out into the blood. Our hydrogen ion is going to be kicked out to our lumen and eventually excreted into our urine. Okay, the next way, and probably more important, is going to be using our ammonium ion. So where do we get the ammonium ion from? Well, it's going to be produced by glutamine metabolism that's occurring in the proximal convoluted tubule cells. So ammonium ions are weak bases, and they donate a couple of hydrogen ions if we're talking about our physiological pH. So just kind of showing you what's happening here. For each glutamine, which is an amino acid that's metabolized, that means that it's going to go through the process of deamination, oxidation, and acidification. And it's going to, by combining with a hydrogen ion. So if we look at glutamine going through these processes, combining with the hydrogen ion, what it's going to yield is 2 ammonium ions and 2 bicarbonate ions. The bicarbonate ions are going to move into our interstitial fluid and eventually into our bloodstream. And if we're talking about our ammonium ions, those are going to move back out to our filtrate and will be removed in our urine. So the last thing for this section is bicarbonate ion secretion. So when our body is in alkalosis, we have too high of a pH, we have another type of cell that actually comes into play. So we have intercalated cell type B. And this is also in our collecting duct with type A. And they exhibit a net bicarbonate secretion and help with reclaiming hydrogen ions to help acidify the blood. So basically,